All right, I'm going to move on to our next presenter, Daniel Wu, who was mentored by Fabia Battistuzzi. Would you like to introduce your intern, Fabia?
Of course. So I had the pleasure of working with Daniel throughout the summer. Daniel came to this project with a good programming background, and he was a perfect fit for the project. This is a project that has been many years in the making, and Daniel finally completed it. So I am very thankful for all his help, and you'll hear everything he has been working on in his presentation.
All right, take it away, Daniel.
Y'all see my slideshow?
Yes.
Okay, hello everybody. My project is to build a computational pipeline for large-scale pangenome analyses. This project was done with my mentor, Dr. Fabia Battistuzzi from Oakland University. Why is a pangenome analysis so significant? Well, NASA is interested in searching for ancient and current life beyond Earth. But first, we need to know what life looks like on Earth. Our earliest life forms were microbes, but they are very different from each other in terms of their lifestyles and functions. Understanding microbial diversity is important because it allows us to better understand how life can adapt and survive under different environmental conditions. Therefore, we need to classify the microbes to better understand their roles in Earth's ecosystem. However, the classification of microbes can be challenging because they are very different among species and within one individual species. So what drives this diversity? Genes, which are fundamental units that contribute to the diversity, both among and within microbial species. However, before we dive into microbial diversity, we need to talk about genetic diversity first, which brings us to the concept of pangenomes. A pangenome is the entire set of genes from all strains within a species. There are three parts to a pangenome: core genes, which are genes that are present in all strains of a species; shell genes, which are genes that are present in only two or more strains of a species, but not in all; and cloud genes, which are present in only one strain. A pangenome analysis gives us a better understanding of which genes are shared within each species and which ones are unique. Strains that have a high number of shared genes are likely to belong to the same species, while strains with a low number of shared genes are likely to belong to a different species. Therefore, this analysis allows us to identify strains that were initially misclassified. To achieve this goal, I built a new computational pipeline to automate the analysis of hundreds of genomes with a single command line. This pipeline ensures that no human mistakes are made during the pangenome analysis. We will also ensure that the pipeline is accessible to other users and that the results are reproducible. For this project, I used a variety of tools and resources. First is the NCBI database, which houses vast amounts of genome files. Next is the Matilda High Performance Computing Cluster at Oakland University. This one is especially important because of its high processing speed and large storage capacity. For the actual pangenome analysis, I used the Rory, which is a software tool for pangenome analyses of prokaryotes. Finally, I used the Python programming language to manage files, integrate different tools into the pipeline, and generate statistical results. This flowchart illustrates the pangenome analysis project my computational pipeline performs. I used the genome assembly files and the genome annotation file from NCBI as my input. For the actual analysis, the input files were formatted and the genome sequences were subjected to pangenome analysis. I will skip some details here. For the output, I was able to generate summaries on pangenome statistics, information on gene presence on strains, and I was able to use the outputs to make inferences on strain classification. All analyses were done using the Matilda HPC cluster. Here's a screenshot of one of the Python scripts I wrote. I wrote a total of 4 Python scripts to perform the tasks required for this pipeline. Then, after testing these 4 Python scripts, I wrote this Slurm script to communicate with the HPC. First, I wrote the /bash lines to request computing resources, such as the number of nodes. Then, I wrote these commands to call the scripts to run. To test my pipeline, I studied the species Bifidobacterium breve, which is a bacterium that lives in the intestines. There is a total of 50 strains within the species. And each Rory run will report the number of core, soft core, shell, and cloud genes. Additional information on each individual gene, such as its name, annotation, and the number of strains the gene is present in, will also be reported. Therefore, from this information, I was able to infer whether a gene belongs to the core, soft core, shell, or cloud gene category. The genomes of strains were added or sorted based on the sequence length, added from largest to smallest, and then the Rory analysis was run continuously to generate the results. Since the number of the results indicating the number of core genes can be influenced by the order in which the genomes were added, we performed 10 random iterations in which we added the genomes in random order for each. As we can see in this table here, as we move right and the number of strains increases, the total number of core genes decreases. Then we performed statistical analysis on the results from each iteration and then fitted a negative exponential curve through a least squares method. Here's a graph plotting the number of core genes versus the number of strains. As we can see, as the number of strains increases, the total number of core genes decreases. The closed circles represent the results based on sorted order, while the open circles represent the results based on randomized order. And the lines represent the negative exponential curve. According to this table here, run 10, which was the run based on sorted order, had the best fit and the smallest residual sum of squares. This indicates that there were no strains that were misclassified, and all of those strains belonged to my species, Bifidobacterium breve. In this project, I learned the concepts of pangenome analysis. Additionally, the emphasis on working in an HPC environment allowed me to learn how to navigate a system based on command line interface. I also learned how to integrate existing software using Python to construct a computational pipeline. This computational pipeline helps identify the core and accessory genes, make it accessible to other researchers, and helps establish a new framework for defining microbial species based on genome content. In the future, this pipeline can be applied across various prokaryotic species and can be used to accurately identify and reclassify misclassified microbial strains to improve our understanding of microbial diversity. Here's a list of the references I used for this project. I would like to thank NASA's Neurodiverse Network program for giving me this internship opportunity to work on this exciting project. Additionally, I would like to thank my mentor, Dr. Fabia Battistuzzi, for her unwavering dedication, patience, attention to detail, and guidance throughout my entire internship. Thank you.
Amazing. Congrats, Daniel, on a great presentation.
Thank you.
That was super clear and a huge amount of work, but it just seems fascinating. And it's also a subject I know nothing about.
Thank you so
So I
much.
was really interested to learn more about it.
I'm open to questions now.
Daniel, I'm going to speak to you directly because this is absolutely fascinating work. It totally resonates with me, and I'd love to talk to you and your advisor about this. We did the same kind of analysis on the core and unique genes of Haloarchaea many years ago, probably when you were just a little kid. And it's really fascinating to be able to decide and determine what actually characterizes a particular type of organisms. So what you've done there is, I'm sure, very, very valuable for the field, and I commend you for what you're doing. So coming at it from someone who has some sense of what you're doing, you did an excellent work. Thank you.
Thank you.
We can take one more question if anyone has one for Daniel.
So how does this help us in the project for finding life and outside of Earth?
Well, you see, the objective of the goal of my project is to analyze a prokaryotic species called the Bifidobacterium breve. By analyzing, and we know that according to my presentation, we know that the earliest forms of life on Earth were bacteria and archaea, which were prokaryotes. Therefore, by analyzing and classifying this prokaryotic species, we can allow a more understanding of prokaryotes in general, which will potentially help NASA find signs of life on other planets.
All right.
Do y'all have anyone more questions?
I think that's it for now. If you have anything more for Daniel, you can pop it in the chat. Thank you for your amazing work.
Thank you.